Manhattan plots are a common type of graph used to illustrate the results of a genome-wide association study, GWAS. For a GWAS, scientists start by collecting genetic data from a large population with varied phenotypes for a particular trait or disease. Then, the scientists look at millions of variants and determine if any of those variants are associated with a particular phenotype of that trait. Typically, scientists will look at single nucleotide polymorphisms, or SNPs, a type of variant where just one nucleotide varies from person to person. For example, to try to find associations between genetic variants and heart disease, scientists would recruit a large group of people to participate in their study, some of whom have heart disease and others who do not. If a given variant is just as common in both groups, it likely has no association with the disease. However, if a variant is more common in the group with heart disease, it may be associated with the disease. Researchers perform statistical tests to identify associations like this. Genome-wide association studies are typically exploratory studies, which give an initial glimpse into variants and genes that are associated with particular phenotypes or diseases. They demonstrate an association between a phenotype and certain variants, but do not prove causation. A Manhattan plot represents all of the variants examined, plotted by their chromosome location. The results of the statistical testing determine whether there is a difference in the frequency of the variant between the test, with disease, and control, without disease, groups. It is called a Manhattan plot because the graph often looks like a city skyline.